In humans, obviously, coronaviruses, not all coronaviruses kill you. 4 of the human coronaviruses cause relatively mild respiratory illness. They just circulate. They cause the common cold. It's estimated that 15 to 30% of common colds are caused by human coronaviruses. There's about 7,000 species of viruses that have been named, but there are way more viruses than that. There are millions of viruses, but in terms of viruses that infect humans, that's a much smaller number. Right now, there's about just over 250 viruses that are known to infect humans. As of now, there are 7 coronaviruses that infect humans, so including, SARS-CoV-2, the new coronavirus. But scientists first learned about human coronaviruses back in the 1960s. We've known about them for a while. The spike protein is, when you see pictures of coronavirus, it's that thing protruding from the outside of it that makes it look like a crown. And so that's why coronaviruses are named coronaviruses. It comes from the Latin corona, which means crown. You can think of them as little machines. They have genetic material, they have proteins, and sometimes they have like a fatty layer around them. And they basically have to get into a cell, and then they take over the cell, they hijack the cell to replicate and to make more of themselves. So when you see emerging pathogens, it usually means that they came from animals into humans. You can be, bitten by an animal. So either a bat could bite you, or mosquitoes. Mosquitoes transfer a lot of virus into humans. You think of all the mosquito-borne illnesses, malaria, chikungunya, Zika virus, dengue, yellow fever, all of those are spread by mosquitoes. People can come in contact with animal droppings or through meat. Viruses have to have specific components to be able to infect certain species. So not just because a virus is in a bat doesn't mean that it can just automatically jump into humans. It has to have certain features. It has to be able to use the right receptor. It has to be able to get into the right cell. But there's definitely potential for viruses that are circulating in animal populations to jump into humans, which is why virus surveillance programs are really important. So viruses can mutate in several different ways. One of the ways is what we call a mutational error. When a virus is making more of itself, when it's replicating, it has to basically make copies of the DNA or the RNA that makes up that virus, that encodes its genetic information. And as viruses do this, the virus machine, the machinery that does it, sometimes it makes mistakes, so it doesn't copy it perfectly, and it can introduce a mutation. An error can really have no effect on the virus, so it can be what we call a silent mutation. It's there, but it doesn't really affect any part of the virus. You can have mistakes that are lethal to the virus, so these mistakes mean that the virus isn't able to grow as well, and so those mistakes, the virus will kind of die out, and you won't see those viruses replicating. But you can also have mutations, mistakes that are introduced into the new virus genomes that help the virus to grow better. Another way that viruses can mutate is through viral recombination or reassortment. When you have recombination and reassortment, what happens for this is you have 2 different viruses infect the same cell. You basically can have information from the 2 viruses be swapped. And so this will be a much larger change than just single little mutations. For the new coronavirus, yes, it's acquiring mutations. All viruses acquire mutations as they grow over time. There have been point mutations that are acquiring, as you would expect, small little mutations as the virus grows over time. That if you didn't see those, you'd be worried. That's how it works. But there is some evidence suggesting that before the new coronavirus jumped into humans, there were some big recombination events that allowed the virus to then be able to infect humans. So So yes, but viral mutation is not something, it can be used as kind of like a fear-mongering or to be like, oh no, mutations, but like viruses mutate, that's what they do. And that's part of how they grow and how they replicate. There's whole groups of scientists that are doing this. They're basically mapping where mutations arose, and it's like putting together a puzzle. So if you isolated a coronavirus sample from a patient in New York City, and you get the whole sequence of the genome, you can then compare it to other sequences that have been isolated from other patients, and you can see what it's most similar to. And so what scientists have found in New York City is that most of the viral sequences, so the genetic information from the viruses in New York, look like ones that originated in Europe. And so you can build a family tree to sort of map where the virus spread. It would be great if we could find the patient 0 who was infected from an animal species. I don't know if that's possible. It can be difficult. You can learn a lot about the science of coronavirus doing that, and I think it's an important goal. One of the things that can get sort of lost in the scientific pursuits, though, is you do want to protect the identity of patient 0, especially in an outbreak like this, because it's not the patient's fault, and so you don't want to have have the patient be ostracized. Age seems to be the biggest risk factor for severe cases of COVID-19, especially as you get older. The complications and mortality rises drastically. Also, health complications like diabetes, obesity, any sort of cardiovascular issues, asthma, those are all risk factors that make you more susceptible to severe disease. But as to why, two people the same age, one person has severe disease, the other doesn't, we don't know yet. A lot of it could be related to the person's genetic information and how their immune system functions. Another possibility is how much virus you're initially exposed to, the dosage, but there's still a lot of unknowns. For a coronavirus, let's say, we don't have any vaccines for human coronaviruses yet, but for a hypothetical vaccine that's being developed, it would contain an antigen, so a small piece of the virus that's specific to the new coronavirus, to SARS-CoV-2. And it would deliver this antigen to your body, but in a way, it's not actually giving you the virus. It's just giving you a small piece, a small antigen of the virus that your immune system can then recognize. So even though you're not infected now, that memory is there, so that if you are exposed to the virus or the bacteria at a later time, it doesn't take weeks and weeks and weeks for your body to mount a specific immune response. Yes, you can. You can get the same virus twice. It depends on the virus. So with the human coronaviruses that are not severe, the ones that cause the common cold, you can be reinfected multiple times throughout your life. I mean, we all get the cold many, many times. In terms of the new coronavirus, it's just too early. We don't know how long you will have protection after you've been infected the first time. There are tons of groups, tons of labs that are working on understanding that right now. If you look back at the original SARS virus, so SARS coronavirus 1, the outbreak that occurred in 2002 to 2004, from people who survived that infection, it seemed that they had a protective response. So that means that they would likely not be reinfected for 2 to 3 years post-infection is kind of how it was estimated. That has given people hope that for the new SARS coronavirus, if there's a vaccine or if you're infected, you will have some protection. It might not be lifelong, but that will protect you for a bit. But there are a lot of viruses out there that are really important for maintaining our ecosystems and helping to control bacterial populations or algae or a whole bunch of other organisms out there. So viruses are critical to life on Earth as we know it. There are a whole bunch of different labs working to figure out how to safely engineer and use specific viruses to target and kill cancer cells in cancer patients. So it's a really exciting area of research in virology, using viruses for good. Another way that viruses are used in the clinic is as vectors. So for viral vectors, what virologists and genetic engineers have done is, instead of just having the virus genetic information in the genome, they swap it out with information they want to deliver. So if you've heard of gene therapy, trying to deliver correct DNA in a human patient. So you think of people who have inherited genetic diseases. So like cystic fibrosis is one that's really common. There's a whole bunch of common and rare genetic diseases. One way to try and treat that genetic disease is with gene therapy. So providing the correct genetic sequence to basically fix the mistake that's encoded in the human genome that gives you the disease. So that's a really exciting way that viruses can be used and manipulated for scientific, like basic science research, but also in the clinical laboratory.